All right, welcome. Today we're going to talk about early life adversity, alcohol, the gut-brain axis, mental health, and relapse. This is where we're headed. This is the final model. I'm going to do some reverse engineering to take you through some of the steps. So just know we're going to build this out. Primary psychosocial exposure is early life adversity, a broad range of stressors, traumas, and deprivation in the first 18 years of life. The most common instrument in humans is the ACE measure. We know that early life adversity predicts alcohol consumption and alcohol use disorders over the lifespan. Multiple associational studies show dose response patterns. And with respect to immediate consumption of alcohol following adversity, we have evidence in animal models. We know that early life adversity links to our outcomes of interest, which are anxiety, depression, and craving. And this has been well described. And we also know that early life adversity associates with several of the mediating pathways along the trajectory, including the gut microbiome, intestinal permeability, inflammation, and the HPA axis. So you can see our antecedent, early life adversity, driving alcohol, gut, intestinal permeability, inflammation, anxiety, and depression. The green boxes we're going to talk about much less, whereas the blue boxes are going to be a focus. We know that alcohol is converted to acetaldehyde and creates reactive oxygen species. We also know that ethanol alters the microbiome, all of which, together can lead to a leaky gut. We know that gut dysbiosis, which is a loss of beneficial bacteria and an increase in more pathogenic strains, also can lead to intestinal permeability, and this relationship is bidirectional. We also know that other metabolites produced by gut bacteria have a lot of important implications for these outcomes, but we will not be getting into those details today. So we've drawn out the link between alcohol, anxiety, depression, and craving. We've recognized that gut dysbiosis can also lead to anxiety, depression, and craving. And we've identified intestinal permeability, aka leaky gut, as an important pathway following alcohol consumption. We also know that disruption of the gut barrier can lead to higher levels of anxiety, depression, and craving in alcohol-dependent subjects. So this was a big study published in PNAS, which really informs much of the research here. An interesting finding was that those with gut leakiness had higher anxiety, depression, and craving even after they spent 2 to 3 weeks being sober. So we know that there's an association here between leaky gut and anxiety, depression, and craving among those who drink alcohol, not necessarily exposed to early life adversity. And what we're going to do next is try to see how we can parse out this pathway. We know that the immune system is involved, that when gut becomes leaky, the intestinal barrier becomes permeable. Microbial products such as lipopolysaccharide, which are endotoxins, can leak through, and this can cross the gut barrier to activate toll-like receptors and essentially start the immune process of signaling for cytokine production. So we've added LPS, gut-derived immune activation, the immune response, toll-like receptors, and then further production of cytokines. It turns out that our knowledge of alcoholic liver disease has progressed in recent years. We understand it now in a way that we didn't, thanks to gut microbiome research. Like I said, this leaky gut leads to leakage into the bloodstream, and these bacterial products actually end up in the liver, where there's a whole new activation of inflammatory cascades, including hepatocytes and Kupffer cells. So liver inflammation can actually lead to peripheral inflammation, further increasing cytokine production. We know that peripheral inflammation can occur particularly related to the lamina propria. It's one of the layers of the mucous membrane, which is associated with the immune system, produces more cytokines, and I'm going to list them in the diagram. And so we have this further systemic inflammatory response. So we get cytokines and other inflammatory markers leading to low-grade systemic inflammation. And where this starts to get interesting is at the level of the blood-brain barrier. We know that alcohol itself might have an impact on the blood-brain barrier. This is the molecule ethanol. But we also know that some of these peripheral blood cytokines also reach the brain to stimulate more cytokines at the level of glial cells. This is the microglia and the astrocytes. So we're adding the blood-brain barrier. We know that the junctions in the blood-brain barrier, the tight junctions, can also become leaky. So these are the neurovascular endothelial cells. And increased permeability from leaky brain can actually then lead to what is being called neuroinflammation. We know that the astrocytes are in contact with neurons to regulate blood-brain barrier permeability. We know that the microglia are involved. The communication between the two of these is less clear at this time. In other words, which one's leading the communication pathway. But we do know that neuroinflammation can originate from ethanol by itself, as well as the generation of inflammation at the periphery that ends up traveling to the brain. So we've got the astrocytes, the microglia, and the neurons added to our neuroinflammation. We know that alcohol can do damage in that respect. And then finally, this is the big question, which has not been answered. Does the amygdala mediate this pathway between neuroinflammation and anxiety and depression? There's definitely some evidence to suggest that regions of the brain are altered from pro-inflammatory states. A review article proposed that amygdala is the site, neuroinflammation specifically in the amygdala, that causes anxiety and withdrawal with respect to alcohol. So these authors propose that amygdala neuroinflammation contributes to the withdrawal behavior by, creating an avoidance of negative affect. And these are symptoms such as anxiety, depression, which are very common during alcohol withdrawal. So this is the almost full model. We've got this outcome of anxiety and craving and depression. And then we bring in the addiction model posed by Koob and Nora Volkow. And this is the negative affect leading to preoccupation. This is, from craving to thinking to using, and the cycle continues. So this cascade's been pretty well described. It's a repeated process by which the negative affective symptoms recreate the consumption behaviors. And so I'm adding in another pathway here, leading to relapse back to alcohol. So there's a feedback loop, and we're looking at how early life adversity combines with chronic alcohol to cause leaky gut, activate the immune system, cause inflammation in the liver and other places, cross the blood-brain barrier, lead to neuroinflammation, which can certainly affect our outcomes as well as possible regions in the brain. We've talked about the amygdala here today, but I think it's safe to say that the reward pathways related to dopamine have been described. There is literature linking early life adversity to changes in the reward mechanisms. So there's much more that needs to be elucidated. I think there's probably some more neuroscience work here that I did not cover in the talk today. These are our references, and that's my contact information. Thank you.